Hello, everyone. Thank you for taking the time to join us for a very interesting series of talks by our expert scientists here at Nexus. Today's sessions will be focusing in on immunogenicity assessments for immuno-oncology drug and therapy development. Specifically, the lectures will be presented in the following order. An overview of the benefits and limitations of the early immunogenicity assessment tools, a review of ADA and immunogenicity assays for preclinical and clinical programs, the use of in vivo models to measure the immune response for cancer therapeutic vaccine development, and finally, assessment of immunogenicity in the context of oncolytic viruses and cell therapies. With that, I'd like to introduce our panel of speakers today. I am Akil Beg, the chair for today's event. I'm a principal scientist within the Immuno-oncology department at the Laval, Quebec, Canada site. Leading off the talks is Sophie Patton, who is the founder and chief technology officer of Immune Experts, who recently joined the Nexus family earlier this year. Sophie is located in Belgium. Out on the west coast of America in Seattle, Washington is Chet Kane, senior director of BioA and Biomarker Sciences. With me in Laval is Danielle Poirier, who is the director of preclinical services, and Noor Rahman, senior scientist in immuno-oncology. Before we begin, I'd like to quickly set the stage. Our team aims to not only showcase the expertise at Nexus, but also highlight areas where immunogenicity plays a prominent role worth considering when developing current and future anti-cancer drugs or therapies. For today's talks, our experts will focus on the specific areas where immunogenicity can be either beneficial or detrimental for anti-cancer drug or therapy development. Finally, the speakers will outline the many ways in which Nexus can help address these challenges in situations of unwanted immunogenicity. As a friendly reminder, speakers, please remember to unmute your mics before beginning. With the stage set, I'd like to turn the floor over to our first speaker, Sophie. Sophie, the floor is yours.
Thank you, Akil, and thank you for the introduction. So I will give you an overview of some assays and tools used to assess unwanted immunogenicity early on. So as indicated in the introduction, immunogenicity is the ability to induce an immune response. So when we administer a biotherapeutic, it will be taken up by the antigen-presenting cells and presented first to the T helper cells, which will get activated and start to produce cytokines and activate the B cells, who will start to produce anti-drug antibodies. So when we talk about unwanted immunogenicity of biologics, this is referring to the so-called anti-drug antibody responses. And when we look at those anti-drug antibody responses for a series of immuno-oncology therapeutics, we can see that certain biologics, such as avelumab, a PD-L1 blocking drug, results in a relatively low incidence of approximately 4%, while others who are also targeting PD-L1, like atezolizumab, have an ADA response of up to 54% in the subjects. So also when used as a single treatment, for example, ipilimumab induces ADA responses in approximately 5% of the patients, and nivolumab, a PD-1 blocker, in 11% of the patients. But when you give them as a combination therapy, that response increases with up to 8% of the patients responding to ipilimumab, but even much stronger responses towards nivo with up to 38% of the patients responding. So this increased ADA response when using combinations is observed also with the use of bispecifics. So with this new combination treatment, there's also the challenge of increased ADA responses. And this can partially be explained by the mode of action, but other factors such as T cell epitopes can influence this ADA response. And with this in mind, it's really important to assess the immunogenicity risk during the preclinical development. So in order to assess the risk and deselect those candidates that might give an immunogenicity issue later on, at Nexus we have a toolbox of assays to support this risk assessment. So first we have the in silico T Cell Epitope Prediction tools that use the sequence and an algorithm to assess immunogenicity. We also have dendritic cell activation assays and whole blood cytokine release assays. That can be used to assess the immunogenicity of biologics and evaluate the innate immune response. Then we have the T cell proliferation assays, such as CD8 depleted and DC T cell assays and peptide assays. And these are really used to assess that immunogenic potential in a population. And finally, we have the MAPS assays that can be used to study uptake and antigen processing of therapeutic proteins to identify those antigenic peptides. And this brings us to the first poll question. And we would like to ask the audience which of the tools for early immunogenicity assessment are used. So if those who want can now give their answers, please. And we're waiting for the results. Not as exciting as the elections in the US, but still we're very curious to see the answers. I cannot really see the answers now, so I guess we still have 10 seconds to go. Yes, so I'm not sure if everyone can see the results, but the top answer is the use of in vitro T cell assays. And also a lot of the people in the call are using a cytokine release and a combination of the above. So thank you very much for this poll question. So that brings us to the first tools, which are the in silico T Cell Epitope Prediction tools. And this cartoon is giving you an overview of the workflow. So the input is actually the sequence of your biologic. At this point, you don't need any material. And the sequence will be screened for the presence of T cell epitopes using a computer algorithm. In our case, that's NetMHC-PAN. That's an artificial neural network-based in silico algorithm. And finally, that will result in an immunogenicity score. And the graph below is showing you such a result of an in silico assessment of 5 lead molecules, so 5 different variants of the same molecule. And where you can see that molecule 5, which is actually the brown line, has the highest immunogenicity score, while molecules 2 and 3, shown in black and red, have a lower score. And with this relative ranking, you can select those leads with the lowest immunogenicity score, and of course, in combination with other tests and other assays. So in summary, in silico tools can be used for the prediction of class 1 and class 2 T cell epitopes. And this can also be used in a vaccine context. So if you want to identify neoepitopes, for example, you can use the same tools. And it allows to give you a relative ranking of the immunogenic potential of your test molecules. It can also be used as a basis for de-immunization or for optimized vaccine construct design. And the advantages of working with us and the NetMHC tool is that it's completely transparent. So there's multiple publications out there that benchmark the tool. And also all known HLAs are taken into account. And we work directly with Professor Morten Nielsen, allowing a correct and in-depth interpretation of the results. So we will now continue with the T cell proliferation assays. As mentioned in the introduction, when we talk about ADA responses in the clinic, we mean the presence of anti-drug antibodies. But today there is no naive B cell available, and therefore we use T cell proliferation assays as a surrogate to assess that immunogenicity. And also in this assay, it's important to assess the response in a relevant population. Next slide, please. So therefore we select PBMC, so immune cells from 50 healthy donors based on the expression of HLA-DR. And the graph in there is showing you in orange the HLA coverage obtained with the experimental population compared to the HLA coverage in the global population shown in purple. And at Nexalis, we have a biobank of over 1,000 cell preparations, all 4-digit HLA typed and quality controlled for this specific purpose. So for us, these PBMC are one of the most important critical reagents, so the quality and the functionality is really very important for us. In the next slide, you can see an overview of such an assay. The above graph is showing you a CD8 depleted assay that's used for non-immunomodulatory molecules. As in the IO context, most of the molecules have an immunomodulatory function. We mainly use the DC T cell assay. And in such an assay, we first will generate monocyte-derived dendritic cells. We will load them with a test article. We will mature them. We will wash away the excess of test article, not to have that direct interference. And then we will co-culture them with autolog T cells. And after an optimized in vitro culture, we can assess proliferation, for example, using EDU incorporation. That's a non-radioactive alternative for tritium-thymidine that can be read on a flow cytometer or one of the commercial tracking dyes. And then, of course, what's important is the result of such an assessment. And these graphs are showing you an example of such an assessment. Here you can see the result of a 50 donor screening expressed in the graph on the left in the number and percentage of responding donors in the bar chart. And in the plot diagram, you can see both the intensity of the response and the percentage of responding donors towards Avastin. So that's the low immunogenic molecules, a VEGF targeting antibody, and also the results of bococizumab and ATR-107, two molecules that were stopped in late clinical development for reasons of immunogenicity and other reasons. And in the upper, so the strongest response is given with the KLH, and that's our benchmark control. So you can see that all donors respond with a relatively high stimulation index. And that's the kind of results we can give you. So you can benchmark your molecules, you can compare them among each other or compare them with benchmark molecules. And that brings us to another assay. We also offer MHC-associated peptide proteomic assays to evaluate antigen uptake and processing, really to identify the immunogenic peptides in your drug. And in this assay, on the next slide, you can see we generate, I would say, bulk monocyte-derived dendritic cells. They are loaded with a test article. They are matured. And then we can lyse them, and after lysis, we can purify that MHC peptide complex, and we can elute and identify those immunogenic regions and map those and using proteomics. So those peptides identified via MAPS or in silico tools can then also be screened to confirm that these were real T cell epitopes. Because, for example, with the MAPS assay, you will only get information on the antigen presenting level. And for such a peptide assay, we use an in-house optimized enriched fluorospot assay where PBMCs are incubated with the test peptides. And after approximately 1 week of in vitro enrichment, they are re-stimulated on a fluorospot plate, which is coated with an anti-cytokine antibody. Fluorospot is a very sensitive method, so you can measure up to 4 cytokines simultaneously, but you can do this at a single cell level. So in the next slides, I will show you the results of a combined in silico MAPS and peptide assay effort published earlier this year in Frontiers in Immunology. So in this study, we used the combination of the MAPS assay, in silico prediction, and peptide assays to see how these tools align and it showed that by integrating MAPS data into the in silico tool, we could have better performance of the predictive tools. And the next slide is just showing you one graph into that publication. So the reference was on the previous slide, so I would encourage you to read the entire paper. But as you can see in the lower part of the graph, you can see the results of the peptide assay evaluating those hotspots identified via either MAPS only, in silico only, or a combination of both. And the results shown in the lower graph show that those peptides identified by in silico and MAPS were confirmed to be T cell epitopes using the peptide assay, but we also found peptides that were not identified via MAPS, but the updated in silico tool could identify those. So it's really showing that the power of the combination of those tools together is giving you a better predictive value. So in summary, at Nexalis, next slide please, we offer an integrated early immunogenicity assessment solution starting from T cell epitope prediction, dendritic cell activation assays, whole blood activation assays, T cell proliferation assays, and MAPS assays, helping you to select those molecules with the lowest immunogenicity risk, or in case of vaccine candidates, those with the highest and the broadest immunogenic potential. And with that, I will give the word to my colleague Sheikh.
Thank you, Sophie, for summarizing the strengths and weaknesses of early immunogenicity assessment tools. Clearly, these assays have come a long way. I'd like to turn the floor over to our next speaker, Czech, who will provide a fascinating review of anti-drug antibodies and immunogenicity assays in both preclinical and clinical programs. Take it away, Czech.
Thank you, Akil. Following Sophie's covering of our offering for the early discovery and lead selection, I will be talking about more key highlights important to successful ADA immunogenicity support of preclinical and clinical programs. In this aspect, I will be highlighting what is important in the life cycle management of the ADA assay, as very often we can have a client that coming back to us after 6 months, 7 months, realizing maybe they need to do additional characterization that was maybe too late. As it had been well documented and published, the unwanted immunogenicity of biologics, it's multifactorial, covering the molecule characteristics to the drug product composition, to the design of a preclinical and clinical program, including the targeted population. Next slide, please. In regard to the, we have a slide before that. Okay. Sorry, yeah. In regard to the molecular structure, have an impact on the ADA assay design, especially complex scaffold or antibody fragment that are potentially reactive to pre-existing antibodies. As an example, I will talk about it in later slide. The targeted population and the geographical distribution of the clinical trial, including the demographics, will impact also the way you'll be getting your commercial sample, and definitely that will impact on the analytical portion of the assay, as well the timeline of your assay development. Any change in the CMC is one of the hot topic, where most often we'll have a client where they have a CMC changes or CQA changes in their drug product, right, that have not been communicated to their CRO, where that will have a big impact on the ADA immunogenicity assessment. Therefore, it's always important when you have a change in your CMCs and CQAs to communicate that with your CRO to make a strategic assessment what needs to be done in regard to reassessing the impact of that. Next slide, please. In regard to the preclinical studies, in general, based on the ICH 6 guideline, you don't need to test your ADA assays in your preclinical until you see some alteration of the PD marker or any change in the exposure, or if you see evidence of immune-mediated reaction. But nevertheless, it's always important to collect the ADA time point and also to have the assay ready in order to be able to test it whenever you see those situations. Next slide, please. For this slide, we must highlight here, it's just the evolving process from the FDA standpoint, where your assay can be started from preclinical. It may be fully or partially, I would say, or fully characterized assay. But as long as you need to have a fully validated assay, well-characterized assay during your application, during pivotal studies. That can be in oncology indication, can be your phase 2B, if you have fast track, or going to be your phase 3 studies for pivotal submission. Next slide, please. We got to overall strategies. Right? When you're dealing with a protein or antibody that is intact protein, the standard tier 3 assays will be recommended, where you have a screening, confirmatory, and titering, and then using the whole molecule for the confirmatory step, that will be the specificity assessment. When you're dealing with immunoconjugates, can be pegylated protein or bispecific molecule, where you have complex molecule, it's always good to have a discussion with the CRO in this situation next list, where we're defining what needs to be done in the characterization steps of different fragments of the molecule. In regard to the peptide, that can be part of the biologic in general, but as due to their size, that can be challenging to generate a specific reagent against the peptide, especially the positive control. Therefore, it's always good to have a very early discussion in assessing what can be done with your peptide assay. Next slide. One of the key challenges, and I categorize them in three buckets, but we have a lot of other challenges, but this one is the main one that may impact how you do your label and also impact the quality of your assay. The drug tolerance, mostly in the oncology indication, due to the frequency and the high dosing that we're giving. We have a lot of circulating drugs that impact your AD assay, therefore having impact of getting some false negative samples. The pre-existing antibodies that can be based on the scaffold of the molecule, in situation the nanobodies and fragment, where definitely due to the Hindi region exposure, you can have a naturally occurring pre-existing antibodies that are reacting to that molecule, or in the situation of oncolytic viruses, where due to the previous exposure to other type of virus, and so you can have a pre-existing antibodies. And the importance of this pre-existing antibodies is critical, as it impacts really the way you assessing your baseline for samples. Therefore, you want to have appropriate cut point for your AD assays, and also due to the underlying discussion of the impact of antibody in the treatment emergent ADAs. The other bucket going to, sorry, next, the other bucket going to be just very quickly, it's the combination of some of the soluble molecules that can bridge or impact the way you assessing your baseline. Next slide, please. In regard to the previous antibodies, as I said, there can be different category, but the main category going to be some naturally occurring previous antibodies, definitely due to different variety of reasons. And we have some that may be mostly specific to the scaffold in the situation of nanobodies, fibr fragment. And then it's always important and critical to make assessment of previous antibodies in your immunogenicity overall assessment, as it will be part of your label and it will be part of your overall criticality assessment. Next slide, please. For the previous antibodies, as I said, it can be naturally. The way you're dealing with them, you have a different way, but you have to assess where they are and what the importance of the previous antibodies. When you start talking about previous antibodies issue, as we know, for our AD assays, our threshold of the screening cut point is to have 5% false positive. Whenever you have higher than 5%, you start having a question, assessing if you're dealing with the issue and how you'll be dealing with that and how to mitigate the issue. And then also one of the key aspects of the previous antibody going to be the level of signal, right? If your baseline is, example for AD ECL assay, you're expecting around 100, 140 ECL values, and you're getting sample that's giving you 500 to 1,000, that you have an issue and you need to assess that. And also definitely the distribution of the signal. As you know, our AD assays, we are recommended to have around 50 for clinical, 50 commercial samples. If you have 30% of your samples that giving values that is maybe 2 times, 3 times of the baseline that are expected, therefore that can be an issue. And then you have always to differentiate the previous antibody to other assay interference, as it's good to have that understanding in order to have a strategy in dealing with them. Next slide, please. In regard to the previous antibodies, the way we can deal it in different ways, but the main one going either you integrate that in your ADA overall immunogenicity assessment, where you say, okay, I'm dealing with the previous antibodies. I have higher than 70% of my sample that's showing previous antibodies. I will not be able to knock it. I don't have an irrelevant molecule to knock it. At the time when you integrate that, I think you need to have a baseline assessment based on previous antibodies, and then in order to differentiate the treatment emergent ADA with a type of factor also, that can be a discussion with Nexalis when you come to us. Or if you have irrelevant molecules that are existing that will not be sharing same CDR, right? If it's your drug molecule that can be used as an inhibition molecule for the previous antibodies. In that situation, just to be careful about the impact on the normal biological effect, right? You don't want to knock your signal completely to the zero banana. You're no longer having biological variability in your sample, and therefore will impact the overstating your cut point that will be assessed. And also, definitely, the last point that need to be taken care of is the impact on the cost and additional steps in your assay. Next slide, please. This last slide is just to highlight some of the critical discussion we can have with our partners, right? Where you coming to us, you have an issue, it's always good. We recommend you having early discussion with CRO, assessing, and especially Nexalis, assessing what you need, and in order to brainstorm different ideas, how to overcome that. In this situation, the client come to us, they would like to quantify anti-drug antibodies against a small protein. And we had a very short time frame, lead time, and also we defined that we may have a challenges in getting positive control to that protein. The idea was to modify an existing assay, that a quantitative assay of that protein, small protein, and to make it in a ADA immunogenicity assay. Next slide, please. Overall, I will just talk about some of the aspects. It had a lot of technical challenges in order to get to where we are in this assay. We did some different optimization where we look into the carrier of this one is the radioimmunoassays, where when we do the propanol precipitation, we have a cut point that was around 1800, and then we did a lot of optimization, different component, and we come to the final end where the normal rabbit serum with the combination of PEG and also the anti-rabbit, goat anti-rabbit antibody, we were able to reduce that signal to 136. That was great. And since we had a percentage binding of 1.4 instead of 7.9, and also we faced some high CV issues during this program, where we're able to overcome that by using aspiration instead of decanting. Next slide, please. This one is just a summary of the overall assay. There is a radioimmunoassay where we're using I-25 labeled protein, and just a combination, as I said, the rabbit anti-human IgG, IgA, IgM, and the goat anti-rabbit having the PEG on it to improve the accelerated precipitation. And we'll remove the unbound tracer, and we count it in using a scintillation gamma counter. And then the specificity assessment is a combination of confirmatory and titration, definitely, where we have overnight incubation with the protein, the small protein, and then do the titration of the anti-antidrug antibody specific to the protein X. Just in summary, overall, what I highlighted here is just to say it's always critical, I think, to have an early scientific discussion, interactive discussion with your Nextselis partner, and in order to make the assessment what is really relevant for you. Back to you, Akil.
Thank you, Chuck, for detailing the importance of ADA and immunogenicity assays, both in preclinical and clinical programs, and more importantly, for highlighting the many factors impacting immunogenicity. No doubt many of our audience members will find this information useful. Let's now shift gears to our next talk describing the use of in vivo models to measure immune responses for cancer vaccine development. I turn the floor over to our animal model expert, Danielle.
Thank you, Akil. The unwanted immunogenicity in the case of cancer vaccine is also very important to look on, mainly the immunogenicity, again, the non-antigen formulation component. The immunogenicity, again, the selected antigen is in contrary a wanted immunogenicity. Usually, the target antigen selection for cancer vaccine development is still challenging because the antigen expression is in some cases not only found on the cancer cell, but also on different organs that could be critical ones. This is mainly the case for overexpressed tumor-associated antigen, where the antigen expression could bring the risk to mount an immune response against different organs. In the case of the cancer testis antigen, the expression of the antigen is usually mainly located on germ cells, where there is a blood barrier between general system and the organ expressing the selected antigen, except for tumor cancer. In addition, especially for tumor-associated antigen that are self-antigen, the self-tolerance need to be bypassed and could bring another challenge to elicit the wanted immunogenicity. Next slide, please. As the second pool question, do you use any of the tool and assay below? Thank you for the answer. Probably waiting a couple of seconds more. Then the simple and complex tumor model is the mainly evaluation tool used, following by the TILs. It's a good thing to know. To assess the wanted immunogenicity, in vivo, several animal models are available and need to be selected based on the vaccine formulation type, the route of administration, and also the immunotools availability. It's also important for the cancer vaccine development to assess the antigen optimization possibility. Often, mutation could increase the antigen stability and immunogenicity with a decrease of unwanted cross-reactivity with other important and sensitive epitopes in the body. This point is highly relevant because the wanted immunogenicity in the case of vaccine development is mainly CD8 T cell response with cytotoxic activity to attack the cancer. In addition, the epitope screening could be very helpful for better understanding the immune repertoire and following the wanted immune response. At Nexcelix, antigen-specific immunogenicity assessment is a part of our work on a daily basis. Usually, we measure the immunogenicity by flow cytometry with different and adaptable marker panel. We are looking at the cell functionality by intracellular cytokine staining after specific restimulation, proliferation, cytotoxic capability, which one could also be assessed by ELISpot. Next slide, please. As tumor animal model to assess a cancer vaccine or therapy with solicitation of the immune system, it's very important to use immunocompetent animal model. These could be divided in simple and complex model. The simple model represents a syngeneic model with implantation of the tumor on the flank of the animal to measure tumor growth and the tumor infiltrating leukocyte. This model allows the target antigen validation, but is less representative of the tumor microenvironment and requests often more than one tumor model to get a better idea in terms of usual infiltration. The simple orthotopic tumor implantation could be followed by imaging system with fluorescence or luminescence, like as IVIS system, and this model is more representative of the tumor's microenvironment, but is still not human. The complex model requests the use of humanized mice. This model allows the possibility of patient-derived xenograft implantation and cell-derived xenograft implantation. This is a more and more important model to determine the efficacy of the vaccine against human epitope and predicted the clinical immune response, including ADA discussed before. Next slide, please. The humanized CD34 mice model is a model of choice. Shortly, this is a model of engrafted human CD34 cell from the human cord blood after the mice irradiation. It took approximately 12 weeks to get a good engraftment, and usually there is the presence of hematopoietic cell subset, like as the B cell, T cell, NK cell, DC monocyte, and macrophages. Next slide, please. The immune cells previously mentioned are important due to their implication into the tumor microenvironment that could be anti-tumoral environment or immunosuppressive environment. The innate immune cells are critically implicated into the tumor microenvironment. This is the cell that define by the cytokine release the immune cell function. Tumor-associated fibroblasts also secrete cytokine into the tumor microenvironment and contribute often to the CD8 T cell exhaustion. Then, the strong immune response elicited by the cancer vaccine against a specific antigen is often not enough to eliminate the cancer and bypass the effect of suppressive cells. In addition, CD8 T cell infiltration and their cytotoxic activity could be blocked when cells infiltrate the tumoral microenvironment. The quantity of MDSC present into the cancer microenvironment and the CD8 on M2 ratio are interesting readout to understand the microenvironment status, pro-tumoral or anti-tumoral. However, different TILs patterns are associated to different cancer type and the correlate with different prognostic. then more complete TILS readout is used for global tumor infiltration picture at Nexcellis. For the murine immunocompetent tumor infiltrating leukocyte, we currently verify the cell infiltration using 3 different antibody panels, looking at the T cell, B cell, and NK cell with separation between the Treg and the CD8 T cell as well, myeloid-derived suppressive cell and dendritic panel that also separate eosinophil macrophages and monocyte, and tumor-associated macrophages panel to mainly identify the polarization of the macrophages between M1 and M2 to get the balance. On the next slide, for the humanized mice panel, we have a T cell, B cell antibody selection with the separation as well between the Treg and CD8, and the myeloid panel that observes the phenotype of monocyte MDSC, DC, and macrophages polarization. Next slide, please. As previously mentioned, the T cell exhaustion could be a problem for cancer elimination. This statue decreases the capacity of the T cell to proliferate and secrete cytokines. At Nexcellis, we have the MCLV mice model that gives the possibility to evaluate the rescue of the immune system with an antibody immune checkpoint blockade, for example, and to evaluate the synergy that could occur when there is a combined treatment. As a readout, we are looking on the cell functionality and the immune checkpoint phenotype, both by flow cytometry. On the next slide, please. In this study case performed at Nexcellis using the LCMV chronic infection mouse model, it was possible to observe that antibody blockade helped to rescue an immune proliferation capability on the left graph, brown dots, compared to the treatment alone on the left graph, red blot, and the rescue of CD8 functionality on the right graph with the synergized action by co-administration of biological treatment on the pig dots. Put all together, the animal immunogenicity, the tumor growth model combined to the ex vivo immune readout and our precious tool in cancer vaccine development. At Nexcellis, we have in place adaptable assay that help and answer clients' need. And thank you for the attending, and I will let the place to Nura.
Thank you, Danielle, for delving in detail aspects of vaccine development and the importance of choosing the most relevant in vivo model. Our final speaker, Noor, will discuss immunogenicity in the context of oncolytic viruses and cell therapies. Noor, please proceed.
Thank you, Akil. So Daniel gave an excellent overview of the immunosuppressive nature of the tumor microenvironment. Since the reasons for the immunosuppressive nature of the microenvironment is multifactorial, we also need to approach the tumor treatment in multimodal fashion. So in addition to the traditional radiation and chemotherapy, we can personalize the treatment for cancer using oncolytic virus, immune checkpoint blockade, and also adoptive T cell therapy. So nowadays, oncolytic viruses can be designed to be specific for tumor T cells, the tumor cells. So once they are inside the tumor cells, they proliferate and also prevent the downregulation of MHC class I molecules. So ultimately, when the virus kills the cancer cell, that will lead to the release of new antigens. And these new antigens can be picked up by mature DCs and then activate the T cells. And those T cells will loop back and kill the cancer cells. And in terms of adoptive T cell therapy, we can introduce CAR T cells, CAR T cells, and also in vitro expanded T cells on new antigens to the mixture. Tumor microenvironment is notorious for exhausting activated T cells. So those T cells can be rescued from exhaustion using immune checkpoint blockade. So these are basically anti-PD-1 and anti-PD-1 ligand antibodies. There have been reports where even in the presence of immune checkpoint blockade, those T cells were not released from exhaustion because the monocytes were picking up those immune checkpoint blockade. Next slide, please. So at Nexcellis, we can determine if those immune checkpoint blockade antibodies are bound to the cells. So we have two models currently, so the bound receptor assay and the free and total receptor assay. So the cartoon on the left shows that the bound antibodies on the surface of the T cells are picked up by anti-drug antibodies, which are marked in violet with the green fluorochromes. And in the free and total receptor assay, we gain more information because we use an antibody that does not compete with the drug. So that the antibody is with the red fluorochrome that marks all the surface exposed receptor, and then stained with another antibody, which is with the yellow marker that does compete with the drug antibody and reveals the free sites on the surface of the T cells. So using a ratio of the free to total sites, we can determine the proportion of the receptors that are bound on the surface of the T cells. Next slide, please. Well, at Nextlis, we can also characterize the oncolytic virus-led killing of the target cells. So basically, the most simplest one is we overlay the cancer cells with the virus and then look for apoptosis by flow cytometry with sample as simple as NXNV and propodium iodine staining. We can also stain with caspase-9 to determine the apoptosis signaling pathways. The other thing that this simple assay can be done is to modify it to determine if the patient has pre-existing antibodies for the virus in the system. So the patient's sera is diluted on a plate in step 1 and then incubated with the virus in step 2 for an hour. And the sera and the virus is overlaid on the tumor cell for 6 hours. And then we can check for apoptosis by flow cytometry. So if the patient has pre-existing antibodies that prevents the virus from getting into the target cells, then the cells will not undergo apoptosis. And we can also assess anti-tumor responses of the T cells in Nextlis with the ELISpot and killing assay. So these T cells can come from PBMC derived from the patients, the CAR T cells, and also T cells that have been expanded in vitro on new antigens. So these T cells can be incubated with the target tumor T cell lines and then demonstrate efficacy of the T cells. So in ELISpot, this can be done with the interferon gamma and granzyme, which will indicate the presence of the effector T cells killing the targets. But to drill down to this individual specific population, we can use flow cytometry. And with InUSight, we can observe real-time killing of the target cells. And we can also titrate the effector to target ratio to generate killing curves. Next slide, please. Using ELISA, we can also determine anti-transgene responses at ELISA. So these assays are used in clinical testing and exploratory work, and these assays can be designed for custom design for that client, or also we can use commercial plates. So in CAR T cells, because the CAR scFv is foreign in nature to humans, so the host can generate anti-CAR responses over time. And those anti-CAR responses will block the efficacy of the CAR T cells. So using CAR exodomain and patient sera, we can show by a simple ELISA assay or even on MSD that the patient has anti-CAR responses that's preventing the CAR T cells from going about killing the target cells. Next slide, please. So Daniel had given us the full panel of the full list of the panels that can be deployed in clinical settings, but these panels will be validated and qualified for fit for purposes for clinical settings. And we can have panels deployed on PBMCs, bloods, and even tumor biopsies. And we can develop T cell, NK, and D cell panels. So we can have T and NK cell activation panel, memory panel, exhaustion panel, and also functional panel based on cytokine, and the DC maturation panel with MDSC panels. MDSC panel to have the different subtypes of the monocytic and PMDC MDSCs. And we can add beads in the panel to count the absolute number of the TB, NK, DC, and MDSC panels. So the fit for purpose will include titration and FMO instrument setting, and the precision runs, which will include intra-assay, inter-operator, inter-equipment, LOB, LOD, LLOQ determination, and post-staining stability. And with the next slide, I give the floor back to Akil.
Thank you, Noor, for highlighting key assays to assess immunogenicity against oncolytic viruses and cell therapies. In our remaining time, I'd like to take a few minutes for questions. Our first question is for Sophie. Sophie, how do you assess and remove background from CD3 bispecifics?
Yes, thank you for that question. Yeah, so these bispecifics come, of course, with quite some technical challenges because their function is to activate the T cells. So therefore, we need to adapt our procedures. We definitely need to use a DC T cell assay platform, because if we would add a CD3 activator to the PBMCs, everything would be activated. So it requires intensive washing to make sure that there's nothing left of the antibody as such, and nothing left of the, I would say, the original activity. So we really need to adapt our procedures in function of specific classes of molecules. And I think everyone in this field working with bispecifics will agree that this is quite a challenging category to make that difference between really the activity of the molecule and the mode of action and the unwanted immunogenicity. And if it's really impossible to do this, then you can go to the MAPS assay or use the peptides instead of the whole molecule.
Thank you. Our next question is for Danielle. Danielle, did you check the reconstitution of the different subpopulations of immune cells in the humanized mice? And if so, was it comparable with the wild type mice?
Thank you. Yes, we look at the different reconstitution of the subpopulation. There is different point that make some difference, depending on the time after the engraftment. There is some population that are not there, and some population. It's the model is a good model to look at the humanized population. The mice population sometime came back with different level, but it's not all the time the same ratio, depending on the time after the engraftment and also the background of the mice.
Thank you, Danielle. Our next question is for Danielle as well. Danielle, to what extent is a viral model applicable to immuno-oncology?
Thanks again. In fact, the LCMV model is a model to look at the exhaustion of the T cell. The same characteristic that is also found in the tumor microenvironment. It's tested to assess the capacity of different candidate or blockade to rescue the T cells capability from the anergic state to the the full capability state. And the goal is not to look with the tumor cell interaction.
Excellent. Thank you, Danielle. Time for one last question. And this one is a question I also am very interested in. It's for Sophie. Sophie, how predictive are these early immunogenicity tools? And do you see correlation with clinical results?
Yeah, I think that's the $1 million question. People that know me know that I personally don't really prefer the word prediction, so we prefer to call it an assessment. And what we see is if we take molecules with, I would say, a high immunogenicity rate in the clinic, we often also see those showing up in our assays. So there is a good correlation for a significant part of the drugs, but we also see exceptions. So I think it's very important to say that, yeah, the human immune system is always much more complex than what we can do in a 96 well plate. So therefore, we would say there is a good correlation, but there's also exceptions. And we like to use this tool more as a deselection tool rather than a predictive tool.
Excellent. Thank you, Sophie. In summary, the Nexus team of experts focused on different areas of immunogenicity and how this plays a role in anti-cancer drug or therapy development. As we heard, immunogenicity can fall into two categories: wanted, as is the case for certain vaccines and IO drugs, where the immune response is beneficial, or unwanted, where the host immune response targets the drug itself through anti-drug antibodies, possibly neutralizing the therapy, or in other cases, anti-HLA antibodies, or even immune rejection. Before we conclude, a quick word about Nextlis. We have extensive experience in all aspects of drug development, from early phase straight through to phase 3 clinical testing. This includes target identification, lead selection, along with both preclinical and clinical testing, making Nextlis your desired partner from safety right through to efficacy screening. With that, I would like to thank our speakers for providing very interesting discussions on various challenges and solutions for immuno-oncology drug and therapy development offered by Nextlis. Thank you everyone for taking the